I'll give you a little bit of background. So I'm the leader of the CPSI Biostatistics program, and I'm also the director of the Department of Medicine Statistics Core. So as a biostatistician, I help people write lots of grants. So in a typical year, I'll help between 50 and 100 grant applications and review many, many more. So seeing lots of mistakes and lots of high quality grants, and I'll try and give you somewhat of an idea of what separates the two. Okay, so the overview, I'll talk some about the key sections in the approach, the preliminary data, the study design, and since I'm a statistician, I'm going to focus on the sample size and statistical method section, and then talk a little bit about general writing strategies. So for the preliminary data section, your goal is to be able to help the reviewers answer this question. Is there a reason to believe that the study hypotheses could be true and that the research team is capable of carrying out the study? So you want to sort of hit both of these points, not only the sort of background, but also to demonstrate your qualifications for actually carrying out the study. So in the preliminary data section, if this is going to be an R1, the strong and relevant preliminary data is absolutely essential to support the application. For an R21 and R03, particularly for an R21, the guideline is that you don't need preliminary data, but that's not true. They say that, and they tell reviewers that you're not allowed to critique the grant for not having enough preliminary data, and then all the reviewers critique the grant for not having enough preliminary data. So, but they just change the words around a little bit but essentially critique it all the same. Some of the key things that you want to have in the preliminary data section: demonstrating expertise with the assays, demonstrating that the assays that may be used are actually that actually work in the patients or samples to be collected, and of course support for the hypotheses. And my guidance is to use figures and tables where all possible. When somebody's reading your grant and they see a lot of text, they're very unhappy. When they see figures and tables, it's much more of a pleasure to read. That being said, what are some of the problems that I see fairly frequently in the preliminary data sections? The first one is insufficient annotations on those figures and tables. Sometimes people don't put axes on the figures and tables, or make the font so small that you can't even read the axes. So if people need to be able to glean the information you want them to from the figures, and so they have to be clear as to what they're actually presenting. The second one is something that is more if you have somebody like me on the study section, but I'll very carefully evaluate the analytic techniques used on the preliminary data. And if I see flaws in the analysis, I will give a very large negative on the significance section. It doesn't exactly follow an approach, but I will usually put that in the significance. And so if, the philosophy as a reviewer is, if they can't do the analysis on the preliminary data right, how are they going to do the analysis on the data that they're going to collect? Okay. Some of the other concerns, one is unrealistically strong or naive preliminary results, which often goes along hand in hand with the presenting needle in a haystack results. So a lot of times I see people with very large-scale molecular data analyses present the one thing that actually came out, but don't actually give you an idea of how many analyses it took to even get to that one thing. And so you have to be able to provide a context for how many things you've looked at before you've actually found the thing that you're going to be talking about, and to give confidence that the strength of that finding isn't just because you looked at 10,000 different things. While preliminary data is important, don't put too much preliminary data in, because you really need to get the methods right, and you need to give them sufficient detail so somebody knows exactly what you're doing. And if you spend the whole approach section on preliminary data, it really takes away from the ability for somebody to evaluate whether you have a good plan or not. Okay, for the study design, the primary question is the design of the study appropriate to address the study aims? So some of the necessary elements that you need to include, obviously the overall study design, whatever that may be. You also need to describe the endpoints and clarify, if necessary, and typically it is necessary, how they'll be quantified and what the measurement scale is. So now if you're saying the outcome is whether they're alive or dead, we'll pretty much have an idea of how you're going to evaluate that. But if it's a biomarker, if it's a more complicated clinical endpoint, being very clear of how it's measured and potentially whether that measure actually has validity for the research question that you're interested in. So that's something that it's critical to put in there. It's also very important to describe your population and control groups, and I should say control groups if necessary. One thing I see a lot is people putting in control groups that are not really relevant to the research question. So there's this idea that every study has to have a control group, and many times that's true, but if you're looking for, say, biomarkers for response to therapy, people who don't have that therapy are probably not going to be really relevant to your research question. So control groups, yes, but only if they help you answer the research question, the primary research question you're actually interested in. And as with the endpoints, any other study measures you're going to be using, it's important to describe the measurement process with enough detail so we have confidence that you're actually measuring what you say you're going to be measuring. From my perspective, one of the things that really gives me a lot of confidence about a grant is when I see a really careful description of the study population. Somebody really knows if the inclusion-exclusion criteria are really well stated. We know whether the demographics and the clinical characteristics of the population that you're going to be able to recruit from are actually relevant to the research question. So I review a lot of cancer biomarkers grants, and many of them say that they're early detection, and then you see if they present a table to show that they have late-stage metastatic patients in their population. Well, that's not actually going to be relevant to answering the early detection question. So, making sure that you can demonstrate that the population you have access to, if it's a secondary analysis, or that you can recruit from, is actually relevant to the research question. And being able to present a table with that information is one of the best ways to really convince people that you're ready to go. I've seen a lot of studies where they say, well, we have a huge repository with well-annotated clinical samples with all the relevant demographics, and then they don't show any of those details to actually prove to you that that is actually representative of the right population. So, that's an important element of proof that you can put out, and it's really shows that you've really thought through the project. Other things that you can think about including in the sort of the study design is, if it's a clinical trial, put in randomization methods, include sections on the validity and reliability of your measures. If you're doing some matching, talk about how the matching is actually going to be done. And if the end result is going to be the construction of some kind of prediction model, whether for diagnosis or prognosis, then including validation strategies for those models. That's particularly for sort of an R01 level. There's an expectation that any models you build will have a strategy for their validation. So what are some of the sort of big failure points that I've seen? The first one is where the study design and the population doesn't match the aims. It seems obvious, but this happens repeatedly when I've reviewed for NIH. Another thing is to make sure that there's sufficient time for recruitment and follow-up within the study. It's sort of a feasibility thing, but sometimes you see the sort of obvious defects there that make the study not really feasible to run. It's important to have clarity with respect to the availability of subjects. If you're going to be recruiting them, maybe go to the institutional EHR and sort of prove that those subjects actually exist at the institution. If you're going to be recruiting them from specific clinics, have letters from those clinics saying, yes, we have these patients. This kind of documentation really supports the feasibility of any project. And when we're writing the approach section, limit the technical details of any assays that you're performing. People are very uninterested in learning what concentration of what solution you'll be using for the assay. If it's a well-documented procedure, reference it and move on quickly. Particularly if the reference is has your name on the author list, then they can say, I've done this before, show the reference, and be done with it, and move on to something that's important. Okay, so sample size. This is why people come to my office. They say, how many subjects do I need, and can you help me write a section to support that number? So from the reviewer's point of view, though, the reviewer is going to ask the question, is the sample size sufficient to give the study the ability to answer the primary study questions? So what are the necessary elements for you to prove, or at least give confidence that the sample size is appropriate? So any sample size calculation starts with the endpoints. So what are the endpoints for the aims? Without that, we can't even start with a sample size calculation. Obviously, we need to specify what that sample size is going to be, but the way we need to calculate it is that for each endpoint, we need the following 5 pieces of information. We need to know what is the estimated effect of the intervention or the magnitude of the relationship that you think you're studying. Now, this is a hard one because most of the time you don't know. And you don't know because you haven't done the study yet. So this is a point which you need to have your best guesses. You need to have a good review of the literature to get a basic idea of what kinds of effect sizes would be actually feasible. And often this is a back and forth process where my collaborators will bring me a lot of papers, we'll look at them together, and we'll try and figure out on the basis of the information there what's a reasonable magnitude of the effect that we can postulate for the study. For individual study measures, we also need to know how much variability there's going to be. This is usually pretty easy with finding, if the measure's ever been used before, we can usually find references to give us an idea of how much variability that might have in the population. We need to specify the level of power. We need to specify whether it's a one-sided or two-sided test. The answer is almost universally two-sided unless you have a really good reason. And we also need to specify what statistical test is going to be used. So in a sense, to do the power analysis properly, we have to really think through the analysis plan together to figure out how we're going to analyze the data, sort of visualize what the data is going to look like, what the analysis plan is going to be, and then the power analysis can only follow from there. So the process by which a sample size calculation is done is not, well, just use 10. It's really thinking through all the aspects of the study and how the data will be analyzed to really get a picture of what we're going to do with the data. Some of the other things that we need to include in any sample size calculation, accounting for study dropouts, accounting for multiple comparisons, and often it's useful to evaluate the sort of sample size and power situation over a variety of scenarios to sort of show that even if maybe some of your assumptions aren't exactly true, that you'll still have decent power if sort of within the range of likely values for sort of your effect size and your variability. So what are the ways that what are the things that generate a negative critique from me? One is if the sample size calculation doesn't have sufficient information for me to replicate it. So I'll actually use my software based on what's presented and then validate that sample size calculation is actually accurate. That's what I'd like to do. In 2/3 of the cases, there isn't enough information for me to actually be able to validate, and so I'll have to make some guesses as a reviewer. You don't want reviewers to guess. You want your information to be solid so that they can really follow your logic and come to the same conclusions that you've come to. The sample size, if the sample size isn't based on any preliminary data that you're presenting, then that's also going to raise a red flag. You show that there's a small effect in your preliminary data, and you postulate a large effect for your sample size calculation. That's not going to fly. One other element is that so many grants involve some kind of prediction modeling at this point. In thinking through the sample size calculation, it's not only the magnitude of the effect but whether you have a sufficiently large sample size to support the prediction modeling exercise that you're planning. And you know, that ends up, figuring that out ends up being sort of at the end of a fairly long consultation often. Here's some bad examples that I've extracted from grants, sort of the what not to do list. So one kind of sort of cop-out on a power analysis is to say that someone else has already done the study and found a significant result with a sample size, so therefore I'll use the same sample size. Okay, that is mathematically untrue, that because somebody else got significance with that sample size, that even if you had the same effect, that you're going to have high power. Okay, so don't do that. For those of you doing basic science, saying, well, a lab usually uses 10 mice per group. Okay, sure, but that doesn't matter. What matters is that there's a rationale for why you have 10 mice per group. 10 mice per group may be too many, or it may be way too few, but you need to justify that. You know, people usually think of it, power is more important for human studies, but it's also examined in any animal studies as well. Sample sizes sort of sick. Power analyses are not provided because there's no information which to base them on. So this is a quote. You may not have any really good preliminary data to suggest how big the effect will be, but that doesn't give you an out from thinking through what's the right sample size and what power you might have based on that sample size. So even if your research is highly innovative and there's no preliminary data, you still have to address this point in a clear way. The fourth example basically says, well, how many patients are there available to recruit? And you know, that's an important consideration for any study for feasibility, but that doesn't justify why that's the sample size. Again, maybe that's way too many, maybe that's way too few, but you know, every study should be able to show that you have feasibility, but it isn't a power calculation. My favorite one is the sort of power calculation on the basis of assertion. I have power based on what I think, and so therefore that's it. So this doesn't go through review well. Okay, so I'm going to give you a sort of what I call a good example. That's something I made up, and this sort of brief example sort of goes through all of the pieces of a sample size calculation that you need to include. So I'll go through it as in the different pieces. So a sample size of 38 in each group. Okay, I've stated what the sample size is going to be. Important thing. Will be sufficient to detect a difference of 5 points on the Beck scale of suicidal ideation. So what is my outcome variable? What's my study endpoint? It's clearly stated. And then how big of a difference do I think there will be between my groups? Okay. And then assuming a standard deviation of 7.7 points and a power of 80%, assuming a two-sided level of significance of 5% and a two-sample t-test. So what's the level of variability? What's the power that we're achieving? Is it a two-sided test? And what statistical test is being used? Okay, so based on my list that I gave you, this one sentence includes all those pieces of information. And if I read this in a grant, I could go and make the calculation and confirm for myself that the calculation is accurate. Okay, so it doesn't have to be long, but it has to have all the required elements. Then often we need to provide some further justification, or in this case, accounting for dropouts. So we increase the number to account for dropouts. And then often what we'll do is then go through some explication of why we think that there is a 5-point difference and a standard deviation of 7.7 points. This could be from preliminary data or from prior literature, but some explanation is very useful there. Okay, for the statistical methods section, the question is going to be, are the statistical methods appropriate for the analysis of the data that will be collected? Okay. I generally advise people to write a separate section for each aim and have those statistical method sections incorporated within each aim, not to wait till the end to go through all the methods. One of the goals of the method section should be to give the reviewers an idea of what's going to be done with the data, and how's that going to allow you to make the conclusions about your study hypotheses that you've proposed. We should have analytic strategies for each endpoint. Sometimes you have the same analytic strategies for all of your endpoints, and you can just state that. The method should be appropriate for the type of data. So to write the method section, you really need to think through for each of the variables, what's the measurement scale, what's the data going to look like, and that will allow you, and potentially in consultation with a statistician, to figure out what the right methods should be. Some of the other considerations, the statistical method should be appropriate for the sample size. So an example of this is if we have a small sample size, instead of using a chi-square test to compare a percentage between groups, we have to use a Fisher's exact test. We should include evaluation and validation strategies for any modeling that we do. And particularly if this is going to be a longitudinal clinical study, whether it's a trial or just an observational study, we need to have methods to account for missing data. Missing data will always occur in any clinical longitudinal study, and there's an expectation that you're going to have a plan for how to deal with that. Some of the ways to fail in the method section, one is ignoring key confounders or demographic variables. I'm going to put this in somewhat of a highlight, specifically the consideration of sex in the analysis, males versus females. This is probably the most common critique I see now if you fail to mention that you'll do a sub-analysis by sex. Every grant that I've written in the last year or two where we didn't explicitly say we're doing an analysis based on sex will ding us for it every time. Whether it's a basic science study with mice, whether it's a human study, this is a giant pet peeve of the NIH at this point, and you just make sure to do it and don't forget about it. Every time I've forgotten to include that, I've gotten dinged, 100%.
So, a question.
Yes?
The sample size will be much reduced if you do a male-female, because in some situations the objective is similar. The objective is similar, male and female. So if you reduce the sample size by, you have to state it in advance, or can you say that you will be evaluating the results in both sexes together? Well,
this is a little bit of an open point, and I think different reviewers interpret the sort of guidelines differently. From a statistician viewpoint, basically, the way I usually take this into account is to say that we will collect the information on sex, and that we will use that as a covariate in our statistical analysis, but not necessarily, but not focus it on saying proving the same effect in males as in females, because in some sense that would require us to double the sample size. And I think some of my collaborators have interpreted it in that way, particularly for the more conservative ones who don't want to get dinged on this, and will just say, well, particularly in animal studies, we'll just do the experiment all the way through twice and prove it both in males and females. From my perspective, I think it's enough to say that we'll have appropriate representation of both sexes, and that sex will be an element of the analysis plan, but not that we'll just run the experiment twice. So, you know, the other thing I want to talk about here is that, particularly in sort of more in the bioinformatics world, but that there is this tendency to describe software but not methods or ideas. So I've read a lot of grants where they say, well, we'll take the data and we'll analyze it in one software package, and we'll move it to another package, and another package, and another package, and somehow we'll get a result at the end of that. That's not very interesting, and it doesn't really show that you really understand what's actually being done. It's more important to say, well, this software package will allow us to do this specific thing, which will then, hopefully at the end of it, allow us to make the conclusion in this way. So it's helpful occasionally to describe software, but that's only a tool, and what we want to know is how you're going to use the tool, and is that tool appropriate for the questions at hand. Just like an assay, the tool is not the important detail. Okay, so I'll talk a little bit about writing strategies that I've seen that have sort of succeeded and failed. So one is using the resource and human subject sections to good effect. So there's often you can put a lot of critical details. If you're always going to be space limited. I've never seen a grant where the problem has been to write enough. The problem is always how to fit in everything you need to say. So often for, say, and also we have the animal studies section. Often I'll see people using the animal sections for a lot of details about the animal population, a lot of details, even some of the sample size calculations the animals put in there with just a reference to them in the main section. Anytime you want to talk about sort of the who and the where, put it in the resources and budget justification sections and leave it as much as possible out of the research plan because you can reference it back. And as I mentioned, the referencing methods, I also mentioned overall, the worst thing that you can do is have a page that's all text. Even if you're just describing methods, figure out ways to include figures or tables to really sort of demonstrate the different components of your project. No one wants to turn a page as a grant reviewer and see an endless block of text. If you think about when you're in high school reading a textbook, you turn the page, you see a lot of pictures, you're very happy. There's not that much for you to read. So, and you can often really make things a lot clearer by putting figures and tables anyway. One thing that people have already mentioned, but emphasizing key points, using bold, using underline, and using repetition. If there's something you can't afford the reviewer to miss, make sure that they don't miss it, because all of us who have written grants have had many times where they say, you didn't do this in your application, and it was clear as day in there. It was written, but people don't read everything, and they don't read everything closely. So if you can't afford them to miss it, don't let them. With the writing strategies, thinking through some of the graphical displays, putting together a graphical theoretical framework, putting the experimental design graphically, putting flowcharts for the aims. This can really help a reviewer really understand the grant in a way that big blocks of text won't. So this was a grant I worked on with Dr. Dubin a number of years ago. And this aim was looking at biomarkers in lung cancer, and it was a very complicated aim where we had multiple different study populations, we had multiple different types of samples from each subject, and multiple different assays that would be run on each of the different types of study samples. And I thought this figure was incredibly well done, that it shows sort of the overall questions, the populations, and all the details in this really simple flow chart. This could have been done in a huge block of text, and you would have no idea what they're going to do at the end of reading that huge block of text. But I think after looking at this, it was very clear exactly what we were going to do. This is an example from a grant I worked on with Vivek Shetty, and this one graph sort of combined together some of the theoretical framework for the study at the top, looking at trauma versus psychological outcomes, specifically PTSD, looking at some of the covariates that we're going to looking at, some of the biological correlates, putting them all together in a nice table, so we can, in a nice graphic, so we can really see how it's all fitting together. And then at the bottom was a table that represented the different markers that we were going to look at. And I find that if you're going to have a lot of different things that you're going to measure, a lot of different markers, it's really nice to put them just in a table. You can give a few details, but it's so much easier to read like this than a big block of text just describing, okay, we're going to look at marker A, then marker B, then marker C, then marker D. Looking at this, you get the useful information without being incredibly bored. Particularly for human subject studies, putting in a sort of a timeline of when all the assessments will be done, again, looks great in a table, looks awful in a block of text. This one was one of the worst examples, but I've seen a lot where I blocked out some of the names of the forms they're using to protect the guilty, but they basically had a huge thing where it just showed, okay, we're going to do everything for almost the whole course of the study, and at the beginning we're going to recruit the subjects, and at the end we're going to write our manuscript. So, this took about a third of a page on the grants, and required them to leave out a bunch of relevant details. So, figures and tables good, but use them efficiently. So last thing, as I'll give you the pitch for the help that the CTSI can provide. The CTSI can provide a lot of help when you're working on your grant application. My group in particular provides support on the statistical methods, obviously, and so we can, it's important to come see us relatively early in the grant writing process, and we can help with the study design, analysis plans, sample size, and also finding appropriate collaborators and budgeting for those collaborators. The people have talked already about how important timeliness is in terms of getting started ahead of time. It's from my perspective, the grants that I see that succeed are the ones where there's time for iterative improvement. Your first draft is never going to be good. Even if somebody's the best grant writer in the world, there's going to be a lot of flaws in that first draft. And having time for you to write, talk to people, make changes, rewrite, do that process over again multiple times. That's the way to get a good product. So just in terms of, I've already said this actually. So, but particularly for on grant applications, the main thing to start out early is study design and sample size. This should happen potentially months in advance because it's going to affect every component of the grant, the study design, and the sample size is going to affect the budget and the feasibility. So getting these things done well ahead of time can save a lot of running down a pathway that just won't work. The statistical method sections are often things that can be written near the end because you have to have sort of all of the details in place before a proper analysis section can actually be written.